to Chapter 13, which covers diversity of microbes, fungi, and protists. This is the first in a series of a few chapters where we're going to be exploring the variety of organisms that exist within the tree of life. Eventually, we're going to get to a couple of chapters where we talk about plants and animals, but in this chapter, we're focused on microbes. And because there's so much overlap between this chapter and Chapter 17, we're also going to be looking at part of Chapter 17 in this lecture, but only the first section. There will be a whole other lecture dedicated to covering the rest of Chapter 17, but we're going to start with Chapter 13 on diversity of microbes. And a good place to start off in our look at this topic is to ask the question, what are microbes? Microbes, also known as microorganisms, are living organisms that cannot be seen with the naked eye. The world around us is teeming with microbes. They live on our bodies, they live in our bodies, they live in our homes, and in just about every environment on planet Earth. And although microbes are small, they are an extremely diverse group. Just because we can't see them doesn't mean they're all the same. There are actually more species of microorganisms than all of the non-microscopic species on Earth put together. And to wrap our heads around some of this diversity, I'm going to show you a simple representation of the complete tree of life, which shows you the major groups of organisms that exist on Earth. So you can see that this tree has main branches that are color-coded. In blue is bacteria, in red is archaea, and then the brown is eukarya. These three major branches are known as the domains in the tree of life, and they represent the broadest categories that biologists recognize in the living world. These domains were established based on evidence from molecular biology, so in other words, DNA sequence data that shows which organisms are most closely related to each other. And then as you can see, each major domain branch has smaller sub-branches that represent more narrow groups of organisms within that broader domain. So, if this illustration captures all of the organisms on Earth, which of these are microbes? Which ones are we concerned with in this chapter? Well, actually, the better question is, which of these are not microbes? Because, in fact, almost every group that you see in this illustration is a microscopic organism. And I'll tell you where the non-microscopic organisms are. Some animals are not microscopic, some fungi are not microscopic, and some plants are not microscopic. Every other branch that you see here is made up of microbes and only microbes. So hopefully this puts in perspective how humongous and diverse of a category microbes really is. And of course, it's beyond the scope of this class to do a deep dive on every branch here. So instead, we're going to tackle some of the broader categories. And we'll start with the two domains that consist of nothing but microbes. And those are bacteria and archaea. These two domains are both made of prokaryotic cells, whereas organisms in the domain eukarya are made of eukaryotic cells. Now those words, prokaryotic and eukaryotic, they take us way back. So let's quickly recap what we learned about prokaryotic versus eukaryotic cells back in Chapter 3. So in Chapter 3, we learned that biologists divide all cells up into one of two categories, prokaryotic and eukaryotic. And we took a look at the images that your textbook shows you for each type of cell, side by side, but we noted that a more accurate depiction of these cells would be for the prokaryotic cell to look like this, because they're much, much smaller than eukaryotic cells. Prokaryotic cells range from 0.5 to 1 micrometers in diameter, while eukaryotic cells range from 10 to 100 micrometers in diameter. And there's also lots of other differences between them, aside from just their size. Prokaryotic cells lack a nucleus. Instead, they store their DNA in a part of the cytoplasm called the nucleoid. Eukaryotic cells do contain a nucleus for their DNA. And in addition to lacking a nucleus, prokaryotic cells lack any other type of internal organelles. They don't have mitochondria, they don't have chloroplasts, they don't have endoplasmic reticulum, or any of those other cell structures that we learned about. Eukaryotic cells do. They contain lots of organelles. And then lastly, we learned that all prokaryotic cells compose single-celled microscopic organisms, namely bacteria and archaea, whereas the types of organisms that are made of eukaryotic cells include plants, animals, fungi, and protists. So now that we remember the differences between prokaryotic and eukaryotic cells, we can get back to this topic at hand, which is looking at the characteristics of these two broad categories of prokaryotic organisms, bacteria and archaea. So let's start with bacteria. We know that bacteria are prokaryotes, but what are their other features? One is that they reproduce asexually, which means that they don't require two partners to mate and contribute genetic information. They basically just clone themselves, which is what you see happening in this video clip here. Another feature of bacteria is that they contain a cell wall made of a material called peptidoglycan. They live in a wide range of environments, including in and on the human body. They live on your skin, they live in your stomach, your intestines, your sweat glands, your hair follicles. And as you probably know, some bacteria are human pathogens. Pathogen is an organism that infects a host and causes disease. However, most are not. Most are harmless or even helpful. Of the trillions of microbes that live in and on your body, many are crucial for your health. They secrete vitamins that your body can absorb, they help train your immune system, and they compete with pathogenic bacteria for resources and space, which helps prevent you from getting sick. However, knowing that some bacteria are good and others are bad means that it's important that biologists be able to identify and distinguish between different species of bacteria to be able to aid in medical treatment. And two of the key characteristics that are used to distinguish between different bacterial species are the shape of their cells and the structure of their cell walls. There are three major shapes that bacterial cells can exhibit. Cocci cells are spherical, bacilli are rod-shaped, and the third shape is called spiral. You can see an example of each of these in the images to the right. The top is bacilli, the bottom left is cocci, and the bottom right is spiral. As far as cell wall structure is concerned, there are two major forms. One of them is called gram-positive, and this describes a cell wall structure that consists of a thick layer of peptidoglycan, while gram-negative cells consist of a thin layer of peptidoglycan in their cell wall. And there's a specific procedure called a gram stain that microbiologists use to dye bacterial cells and view them under the microscope in order to determine the structure of the cell wall. And in that gram stain procedure, gram-positive cells end up being dyed purple and gram-negative cells end up being dyed pink. And this can allow you to distinguish between them to help identify the species. So this will be the topic of our first checkpoint of the lecture. Salmonella enterica is a species of bacteria that often contaminates food and causes infections of the digestive system. In the news media, it's usually just referred to by its first name, Salmonella, so that's probably what you've heard of before. And here you can see an image of this species as seen under the microscope. So for this checkpoint, please tell me, what is the cell shape and cell wall structure of Salmonella enterica? So that concludes our discussion of the domain of bacteria. Now let's talk about archaea. We know that archaea have something in common with bacteria, which is that they're both made of prokaryotic cells. Another thing they have in common with bacteria is that they reproduce asexually. However, beyond these basics, archaea are actually pretty different from bacteria. They do contain cell walls, but those cell walls are made of a different substance that is called pseudopeptidoglycan, not peptidoglycan. Another big difference is that archaea are not pathogenic to humans. So unlike bacteria, they don't cause infections. Many species of archaea are extremophiles, which is a word used to describe species that live in extreme environments. For example, take this little guy, Pyrococcus furiosus. This is a species of archaea that lives around hydrothermal vents at the bottom of the ocean. Hydrothermal vents are places where heat from the molten core of the earth seeps up to the surface, and the water around those hydrothermal vents can reach up to 700 degrees Fahrenheit. So I think we'd all agree that that is a pretty extreme environment. but that is the preferred habitat for this species. In general, there are three categories of extremophiles. You've got your thermophiles, like Pyrococcus furiosus, that live in very hot environments. Then you have halophiles that live in very salty environments. And finally, you have acidophiles that live in very acidic environments. This brings us to our next checkpoint. What type of Archaea would you find living in the Dead Sea, which is a lake in the Middle East that has a salt concentration about 10 times higher than the ocean? So we're finished looking at the two domains of prokaryotic microbes. Now we're going to turn our attention to the eukaryotic microbes. And in this domain, things are a little bit more complicated because eukarya contains a mixture of microbes and non-microbes. And in this chapter, we're just focusing on the microbes. We'll get to the non-microbes later. So let's start by talking about the portion of animals that are microscopic. Some animals are microscopic for all or some of their lives. And despite being microscopic, this doesn't mean that they are just composed of a single cell like bacteria and archaea. Microscopic animals are multicellular, but they just have incredibly small bodies. Other characteristics include the fact that, like all animals, they are heterotrophs. And that's another word we haven't heard in a while. So let's remind ourselves in a checkpoint. Recall from chapters 4 and 5, what are heterotrophs and how are they different from autotrophs? So microscopic animals are heterotrophs, and most microscopic animals belong to one of two groups. Most are either arthropods, which is the name of the animal group that includes insects and crustaceans like crabs, or they are roundworms. So most are cousins of insects, or they're a teeny tiny type of worm. Some examples include Demodex mites, which you can see pictured here as seen under the microscope. Demodex mites live in human hair follicles, especially the follicles of your eyelashes and eyebrows. Another example is copepods. Copepods are tiny aquatic animals that live in saltwater and freshwater habitats. They're almost like super tiny shrimp or lobster. A third example is root knot nematodes. These are a type of roundworm, and they are pathogenic, but not to humans. They're pathogens to plants. They infect the roots of plants and they siphon off their nutrients. And it's estimated that they are responsible for 5% of global crop loss, which is a lot for just one little guy. So this gives you an idea of some of the types of organisms that fall into this category of microscopic animals. Next, we're going to move on to talk about microscopic fungi. The situation here is similar to the situation with animals, where not all fungi are microscopic, which I'm sure you're aware of. If you've ever seen a mushroom, mushrooms are a type of fungus that is clearly not microscopic. But some fungi are microscopic, and the two major categories of microscopic fungi are molds and yeasts. So those are the ones that we're focusing on here. Molds are multicellular microscopic fungi, while yeasts are single-celled microscopic fungi. So you can see an image showing a mold under the microscope on top, and then you can see a yeast collection under the microscope on the bottom. Molds are made of filaments that are composed of multiple cells, and yeasts are just a single cell. And then another difference between these two types of fungi is how they reproduce. Most molds reproduce using spores, and I've dropped a little black arrow in the top image pointing to some of the spores in this mold. They're the tiny little dots that you can see sort of sprouting from the filaments and scattered all around the view. And then yeasts, on the other hand, they mostly reproduce through budding. Budding is a type of cell division where the cell grows a little nub, and then the nub slowly grows into a whole new cell, and then it breaks off from the parent cell. And in the lower picture, I have added a white circle around a yeast cell that you can see is in the early stages of budding. Other characteristics of molds and yeasts include the fact that they are heterotrophs. This is something they share in common with animals. They can't perform photosynthesis, Instead, they have to eat to get their nutrition, but their version of eating is simply absorbing food particles from their surroundings. Fungi also tend to grow in moist environments, although for some species, their spores can be very resistant to drying. And also, some species of molds and yeasts are human pathogens. For example, toenail fungus is caused by a type of mold that feeds on the keratin in your nail bed. And then ringworm, which is what you see on the bottom, is a skin infection that is also caused by fungus. Its name is very misleading. It's not a worm. It's caused by fungus. However, like bacteria, not all fungi are bad. Many are involved in producing foods, beverages, and medicines. For example, Saccharomyces yeast, which is represented on the top, is used in the baking of bread and the brewing of beer. And on the bottom, you see a type of mold called penicillium. Penicillium sounds a lot like the word penicillin, and that's not a coincidence. The antibiotic penicillin comes from Penicillium mold. It's a naturally occurring compound produced by this mold as a form of self-defense against bacteria. And in this picture, the big white splotch at the top of the petri dish is a Penicillium mold growth. And then the streaks that you see underneath are bacteria that are growing in that very same petri dish. But you may notice that there is an empty ring around the mold colony where the bacteria are keeping their distance. And that's because the mold is producing a substance that kills the bacteria. It's producing penicillin. And we harvest that penicillin and use it as medicine. So molds aren't all bad. They can be very helpful. So that covers our look at microscopic fungi. Now let's talk about microscopic plants. Microscopic plants are called phytoplankton. And here is what they look like. This image shows an assortment of phytoplankton under the microscope, and they're actually considered a type of algae. But algae is a big category that also includes larger aquatic plants that are visible to the naked eye, like seaweed. So these phytoplankton are microscopic algae. And like all plants, they contain chloroplasts, and they perform photosynthesis to meet their energy needs. Now, we learned about photosynthesis back in chapter 5. And for this next checkpoint, I'd like you to think back to that chapter and tell me what are the two things that are produced from the process of photosynthesis. When plants do photosynthesis, what two substances do they make? So, now having remembered what photosynthesis generates, it should make sense to us to learn that phytoplankton form the base of the food chain in ocean ecosystems. They capture solar energy and they use it to make nutrient material, and then they are eaten by zooplankton, which are tiny microscopic animals like those copepods that we just recently saw. And the zooplankton are eaten by small fish, which are eaten by larger fish, and so on. So even though you can't see them, the whole aquatic food chain is held up by phytoplankton that channel energy into the system by performing photosynthesis. And it's not just ocean creatures that depend on these phytoplankton, because the phytoplankton are also responsible for generating a majority of the oxygen in the Earth's atmosphere. So estimates vary, but somewhere between 50 to 80% of the oxygen in the atmosphere is generated by phytoplankton, which is more oxygen than what the plants on land give us. So these phytoplankton play a very important role in the grand scheme of our living systems on Earth. So that's microscopic plants. We have one more category of microscopic organisms to cover, and that is protists. We've talked about microscopic animals, microscopic fungi, and microscopic plants, and protists are everything else. Protists are defined as single-celled microscopic eukaryotes that aren't animals, fungi, or plants. And I know that sounds silly, but that really is what this group is. It's an extremely diverse catch-all category. It's a bucket that's designed to capture anything that's not a plant, animal, or fungus and is microscopic. The only thing that unifies this category is that they are composed of a single eukaryotic cell, and they don't belong in any of the other categories. Of course, given the diversity of protists, it means we can't cover the full breadth of all the different types, but I'm going to highlight them here because while most of them are free-living, some of them are human parasites. So we'll talk briefly about a few of the more medically important ones. First, Plasmodium falciparum, which we've mentioned before, is a protist that infects red blood cells and causes malaria, which is a very serious illness that affects hundreds of millions of people worldwide. It's transmitted by mosquito bites, and it's very difficult to treat. And it's so deadly that, as we talked about in our last lecture, it has caused the sickle cell allele to remain prevalent in sub-Saharan Africa because people who have one copy of this allele have some protection against malaria infection. Another protist that's parasitic to humans is Giardia lamblia, more commonly known as just Giardia. This one infects the digestive tract. It is transmitted by drinking contaminated water, and it causes the diarrheal disease known as giardiasis. By some estimates, Giardia is considered the most common intestinal parasite in the US, so it is here with us. And it doesn't affect just humans, it also affects some animals as well. So I once had a cat that had giardiasis, and that was not fun for anyone. The last one that I'll highlight is Trypanosoma cruzi. This is another blood-borne parasite. You can see it swimming around in this sample of red blood cells under the microscope. But unlike the parasite that causes malaria, this one doesn't actually enter and go into your red blood cells. This parasite is transmitted by bites of the triatamine bug, which is also known as the kissing bug. And the reason why it's called the kissing bug is because these bugs are attracted to the carbon dioxide that you exhale. So they tend to bite people around the mouth. And if you're looking at this bug and thinking, those kind of look familiar, I wouldn't be surprised if you've seen them before because they are native to the southern US and they're common to find here in the Sonoran Desert. And there have been studies of kissing bug populations in Arizona that show that up to 50% of them are carriers of this parasite. But transmission is rare because the bugs are nocturnal and most people sleep in sheltered areas that are relatively insect-free. But if you do contract Chagas disease, the early symptoms include flu-like symptoms, rash, swelling near the bite site, stomach problems. If left untreated in the long term, it can progress to heart failure. So those are a few parasitic protists to be aware of. And this means that we have covered an overview of all the different categories of microscopic living organisms. But we're not done yet, because while this illustration shows us the tree of life, there are some microbes that aren't part of the tree of life. Some microbes aren't actually alive. They're not made of cells, and they don't fit anywhere in this broader map of living organisms. And there are a few examples of microbes that this applies to, but the big category is viruses. I'm sure you've heard of viruses before. I'm sure you've even experienced being sick with a virus before. But what actually is a virus? Viruses are extremely small parasites that invade and infect living cells. They themselves are not made of cells. And that's probably why your textbook put them into a different chapter. So this is where we're transitioning over into the first section of chapter 17. We're done with chapter 13 now. So viruses are not made of cells. Instead, they have a very simple, minimal structure. They're made of genetic material surrounded by a protein shell called a capsid. And here's a little cartoon illustration of what a virus looks like. One thing that is interesting about viruses is that their genetic material can be either DNA or RNA. Some viruses have DNA, others have RNA. So let's take this opportunity to remind ourselves of what we learned in chapter 9 about the differences between DNA and RNA. Both of these are nucleic acids, but they have differences in their structure. What are two of those differences? Okay, so we know that viruses are tiny parasites that get inside of your cells. They're made of genetic material stored inside a protein shell called a capsid. And some viruses, but not all, additionally have a layer of lipids outside of their capsid called an envelope. So in this illustration, the genetic material is the little purple string that you see in the middle. The capsid is that elongated hexagon shape that's surrounding it. And then the big circle that surrounds everything is the envelope. And on the exterior of the envelope, you'll see these green knobs sticking out. Those are meant to represent the spike proteins. Spike proteins play a very important role for the virus because they are the part of the virus that interacts with the host cell. So the spike proteins attach themselves to receptors on the cell wall or the cell membrane of the host, and they convince the host to let the virus inside. Now we can see that this virus, it has some things in common with living organisms. It's got genetic material, it has proteins, it has lipids, it seems to be made of the same stuff. So why isn't the virus considered alive? The reason why viruses are not alive, despite some of the physical similarities they have with cells, is because they aren't able to perform some of the main functions that unify living organisms. So remember back in chapter 1, we went through a list of 7 characteristics that are shared by all living things. Viruses lack some of those characteristics. Namely, they cannot reproduce on their own, they cannot process energy on their own, and they can't maintain homeostasis on their own. They do these things, but they are entirely reliant on their cellular hosts for these functions. They take over cells. They invade, they hijack the organelles and the resources of the cells, and only by doing this are they able to perform these functions. But without a host, viruses can't do these things, and so they're not considered alive. And importantly, the process of the virus executing its invasion and takeover is what causes the disease to happen. So let's see what that invasion and takeover process looks like under the microscope. So here in this image, we can see a number of viruses preparing to invade a host cell. So the host cell is the big blob in the bottom right, and the viruses are the much smaller, darker colored structures that are lined up above the host cell. So this gives you a sense of how small viruses are compared to cells. And what's cool about this image is that you can actually see the envelope of the virus, and then inside of that envelope, you can see the capsid as well. So these viruses will invade the host cell, and then they will take over and force the host cell to use its resources to allow them to multiply. They take control of the cell and make the cell dedicate its energy and efforts to creating more viruses. And as you can see in this image, the amount of viruses generated from just one cell can be extremely numerous. All of these little spots now are viruses that have been created inside of the cell. And then those viruses will exit the cell and they'll travel to find a new cell to infect, where the process will then repeat from the beginning. So this is how viruses operate. And I want to take this moment and emphasize something really important, which is a very common misconception about viruses, and that is that viruses are not the same as bacteria. Viruses and bacteria are two completely different things. They often get lumped into one category because they can both cause infections, but they are quite different. Bacteria are microscopic living organisms made of prokaryotic cells, but viruses are microscopic, non-living parasites. When it comes to bacteria, most of them are harmless or even helpful. But when it comes to viruses, most of them are pathogens and cause disease. In bacteria, of the species that are pathogenic, very few of them penetrate and invade your cells. There are some rare bacteria that go inside of your cells, but they are the exception. They're not the rule. Whereas with viruses, Their MO is to penetrate and invade your cells. That's just how they operate. And then lastly, when bacteria cause infections, they are susceptible to antibiotics. Antibiotics are medications that you can take to eliminate bacterial infections. But viruses are not susceptible to antibiotics. They are very difficult to eliminate without causing collateral damage to the host cell, meaning your cells, the ones that are experiencing the infection. So let's talk about why that is. Why don't antibiotics treat viral infections? Well, antibiotics are medications that kill bacteria by targeting and destroying essential prokaryotic cell structures that are not present in human cells. And this last part is crucial because in order for an antibiotic to work, it has to kill the bacteria without killing you. So, for example, the antibiotic penicillin works by destroying the bacterial cell wall. Bacteria need their cell wall, they die without it. Human cells do not have a cell wall, and so penicillin is able to eliminate the infection without harming the person who's taking it. So that makes sense. But when it comes to viruses, you're facing a different sort of challenge. How do you target and eliminate something that, number one, has an extremely minimal structure, so there's not much there to even attack? Number two, is inside of your cells. And number three, uses your cells' organelles and resources in order to survive. And I put survive in quotes because, as we now know, viruses aren't really alive. But if you try to take away or destroy the things that they are relying on to function, then you are, by extension, taking away or harming something that your own cells rely on. So you see how it's a catch-22. And this is why developing antiviral medications is very challenging. It's very hard to create drugs that eliminate viruses without harming your cells. And given that this is the case, the best way to address most viral infections is prevention. Rather than treating them after they've already taken hold in your body, preventing them from happening in the first place is a much more achievable approach. And we'll talk more about the ways that we prevent them in our next chapter on the workings of the immune system. So let's tackle another checkpoint. Colistin is an antibiotic that destroys cell membranes. It's used as a last resort treatment for bacterial infections that are resistant to multiple other antibiotics. And the reason why it's used as a last resort is because it has significant toxic side effects for the patients who take it. Why is this medication toxic to humans? So we've been focusing on the role that viruses play in human disease, and we're going to continue to keep that as our focus. But we also need to briefly mention that viruses can infect all types of hosts, not just humans. All types of cells are susceptible to viral infections. So for example, here we have an image of a bacteriophage. This is a virus that infects bacterial cells. And that too should drive home the point that viruses are not the same as bacteria. Viruses can actually infect bacteria. They can also infect plants. So here's an image of tobacco mosaic virus under the microscope up top, and then some tobacco leaves on the bottom that are infected with this virus. And of course, as you know, viruses can infect animal cells, including human cells. So with our remaining time in this lecture, we are going to focus on learning about a few select animal viruses that are particularly relevant to humans. And those are influenza virus, which causes the flu, COVID-19, and human immunodeficiency virus, or HIV. So we'll start with influenza virus. The influenza virus has RNA, not DNA, as its genetic material. And unlike some other viruses, it does have an envelope outside of its capsid. And on that envelope, there are two main types of spike proteins. H spikes are composed of a protein called hemagglutinin, which is where the H comes from. And these spikes interact with the host cell receptors to gain entry. The other type is the N spikes, which are made of a protein called neuraminidase. That's where the N comes from. And these N spikes send signals to the host cell when they're ready to exit and find a new host. And if these letters H and N sound familiar to you in the world of flu virus, that's no coincidence because the designated strain of the flu virus is determined by the type of H and N spikes that a particular virus has. So for example, you may have heard of H1N1. That is the strain of flu known as the swine flu. It can infect pigs. It can also infect humans. So this graphic right here shows you the various strains of flu and what hosts they can infect. H5N1, another example, that is the avian flu, also known as the bird flu, which primarily infects birds but can also infect humans as well. So the bottom line, the particular type of H spike combined with the particular type of N spike is what makes the flu virus strain and what determines the type of host that that strain can infect. So now let's take a brief look at how the virus, the flu virus specifically, goes through its life cycle and causes infection. The flu virus will invade cells that line your respiratory system. So here we have one of those cells shown in this big orangish oval, and the nucleus of the cell is shown in purple. And the flu virus you can see touching down on the top of the cell and convincing the cell to let it inside. And then once inside, the capsid of the flu virus will break open and release its RNA, release its genetic material, which will travel into your cell's nucleus. And then from there, from the nucleus, which you guys know is the control center of the cell, the RNA will hijack the cell and force it to create more capsid proteins and more RNA copies. So let's pause this journey for a second for a checkpoint. In order to create new capsid proteins, the virus needs to take control of which structures in the cell. So in other words, which structures are responsible for creating proteins? Okay, getting back to the life cycle of the flu virus. The virus forces the cell to make more capsid proteins and more RNA copies. Then those pieces are assembled into new viruses, which get released from the host cell, and then they're free to go find a new host cell to infect. So this is how the flu virus operates. And over time, this process kills the host cell. It kills your cells lining your respiratory tract. Simultaneously, your immune system will also be trying to destroy your infected cells to halt the progress of the infection. And together, these two things cause the symptoms of the flu and all of the nastiness that you experience when you have this infection. So that covers our look at the flu. Now let's move on to talk about COVID-19. COVID-19 stands for Coronavirus Disease 2019. So COVID-19 is the disease, and the name of the virus is actually coronavirus. Coronaviruses have spike proteins that, under the microscope, make the virus look like it has a crown, or in other words, a corona, which is where it gets its name. And like the flu virus, coronaviruses enter and exit the body through the respiratory system. They're transmitted through respiratory expulsions, like coughing or sneezing, or through direct contact with someone who is infected, or through indirect contact. So for example, if someone sneezes into their hand and then touches a door handle, and then you come by and touch the door handle after them, that is called indirect contact. And COVID-19 is not the only disease caused by a coronavirus. There are actually 7 known strains of coronaviruses that have human hosts and many more that have non-human hosts. 4 of these are causes of the common cold, but there are 3 that lead to much more severe diseases. These include Middle East Respiratory System, or MERS, Severe Acute Respiratory Syndrome, or SARS, and then COVID-19. Thankfully, MERS is not very contagious, and SARS has been eradicated, so it's no longer around. So now we really just have to worry about COVID-19 among the coronaviruses. And how contagious is COVID-19? When we're talking about the contagiousness of any infection, there is a value that's used called R0, which is represented by the letter R with a little 0 subscript next to it. And R0 is a value assigned to represent the number of individuals to which an infected person transmits a communicable disease. Basically, it's a number that says, if you have this disease, on average, how many people catch it from you? And so the higher the value, the more contagious the disease. For the flu, this number is 1.5. And remember, these are averages, that's why that's not a whole number. For measles, R naught is 15. Measles is one of the most contagious diseases known to man. For COVID-19, it's about 1.9. So it's a little bit more contagious than the flu. And something you may have also heard about COVID-19 is that it's become more contagious over time. The virus has accumulated mutations in its spike proteins that enhance its ability to invade our cells. So here in this chart, you can see of the different variants of COVID-19. We've got the beta, delta, and omicron, along with their discovery dates on the left. And then we have their R naught values in the right-hand column. And you can see how the R naught value has gone up over time, meaning that the contagiousness has gone up. So in this checkpoint, I'd like you to think back to chapter 11, part 2, and tell me, this is an example of the virus evolving by what mechanism? So that covers COVID-19. We have one more virus to talk about, and that is human immunodeficiency virus, or HIV. We'll start with a little bit about the structure of HIV. So like influenza virus, HIV has RNA, not DNA, as its genetic material. And also like the flu, it has an envelope surrounding its capsid. However, unlike the flu and unlike COVID-19, HIV contains a special enzyme called reverse transcriptase, which turns its RNA genetic material into DNA. The name of this enzyme, reverse transcriptase, it makes a lot of sense based on what its function is. And in this checkpoint, I'm going to ask you why. Think back to chapter 9. When a cell carries out the process of transcription, what is it doing? And then based on this, why does the name of the enzyme reverse transcriptase make sense? So we know a little bit about the structure of HIV. Now let's talk about how it operates. HIV is transmitted from person to person through sexual activity or direct contact with blood. It infects a very specific type of cell in your body called T cells. T cells are the type of white blood cell that activates your immune system to respond to infection. So when you get an infection, it's the T cells that mobilize everything else to start working on protecting you. So that means that over time and without treatment, the effect of HIV erodes the body's ability to fight infections. And this can lead to the onset of acquired immunodeficiency syndrome, or AIDS. However, with treatment, HIV infections can be prevented from progressing to AIDS. AIDS is what happens in a late-stage, untreated HIV infection. And the reason why HIV causes these long-term infections is because the virus integrates its genetic material into your DNA, which makes it virtually impossible to cure. Let's see how this works. Here's an illustration showing an overview of the life cycle of HIV. So we'll start in the upper left-hand corner with step 1, which is where the virus attaches itself to one of your T cells and gains entry. And then once inside, it will release its RNA, and then that special enzyme, reverse transcriptase, takes the viral RNA and turns it into DNA. The DNA then enters the nucleus of your T cell, and it integrates into one of your chromosomes. It actually inserts itself into your DNA and becomes part of your genetic material. From there, your cell will treat the viral DNA like it's any other gene in your chromosome. It will read it, transcribe it, and use it as instructions for making more viruses. And then it will assemble those viruses so that they can be released and find more host cells to infect. And over time, this process kills your T cells and makes it so that your immune system can't respond to other infections, and so small, normally harmless infections can suddenly become life-threatening. Thankfully, though, there are medications that can treat HIV infections. HIV is one of the few viruses for which there are effective antiviral medications available. We talked about how antibiotics don't work against viruses, so antibiotics have no effect here. But the pharmaceutical industry has developed medications that effectively target and treat HIV. Collectively, these are called antiretroviral medications, and they include reverse transcriptase inhibitors, which disable the enzyme reverse transcriptase and prevent the conversion of RNA into DNA. Another class of drugs are integrase inhibitors, which disable the enzyme integrase, which is the one that inserts the viral DNA into your chromosome. And there are also protease inhibitors, which disable the enzyme protease, which is involved in assembling the new viral pieces into complete viruses. And with these antiretroviral drugs, it's important to note that these medications treat the infection and prevent it from progressing to AIDS, but they don't technically cure it. The virus is still in your DNA. However, even though they can't cure the infection, these drugs can get patients to quote-unquote undetectable status, which is the medical term meaning that there is so little active virus in the body that it can't be detected using the tests that we have. So a person who is undetectable tests negative for HIV, even though they still have a latent infection that is being suppressed by the medications. And it's necessary to consistently stay on a regimen of these medications to remain undetectable. If a person has a gap in treatment, then the infection will rear its head again. But if a person is able to consistently stay on antiretroviral drugs long term, then the prognosis is very good. It's much, much better than it used to be, and it continues to improve. And here are some graphs that put this in perspective. The first graph on the left shows the number of HIV-related deaths each year starting in 1987, running through 2011. And these data come from Canada. The numbers would be much higher if we were looking at the US because the US has a much higher population. But what the graph shows is that HIV was causing increasing numbers of deaths and becoming an increasingly serious public health problem through 1995. And then in 1996, which is the bar that they've highlighted in red, this is when antiretroviral therapy, or ART, was first introduced for treating HIV infections. And from there, you can see a steep and sudden drop-off in the number of deaths. So this really illustrates how effective ART is and how many lives it has saved. And as I said, prognosis has been steadily improving over time, such that in 2008 to 2012, the life expectancy of someone who's HIV positive is not so very different from someone who is HIV negative. It's 72.4 years. So this is all very good news. But it's also important to keep in mind that these data come from Canada, which is a rich, developed nation with universal healthcare where people generally have very good access to consistent antiretroviral therapy. The picture looks very different in some other parts of the world where HIV is prevalent, but these expensive drugs are not as readily available or the healthcare infrastructure isn't as strong. We'll talk about that in a moment. But another important thing to note about antiretroviral drugs is that they are also highly effective when used preventatively. So if a person is in a position where they have a known risk of contracting HIV, they can get on these medications to prevent an infection from taking hold. And this strategy is known as pre-exposure prophylaxis, or abbreviated PrEP. PrEP is recommended for HIV-negative individuals who have HIV-positive sexual partners, inject drugs and share injection equipment, or are otherwise at risk for HIV infection. And PrEP is extremely effective. It's 99% effective at preventing HIV infection when taken as prescribed on a daily basis. The problem with this approach, though, is that the countries in the world that are most severely affected by HIV, these are countries that have trouble accessing healthcare on a consistent basis. This map shows the countries of the world color-coded by the percentage of the population that is affected with HIV as of the year 2019. And as you can see, the highest concentrations are in sub-Saharan Africa, where in many places people don't have a local medical clinic that they can consistently access. Maybe they have to travel far distances to see a doctor. Maybe they can't afford antiretroviral medications. So there are additional challenges at getting these treatments to the places where they are needed the most. However, one bright spot has been the release of long-acting versions of PrEP that don't need to be taken as a daily oral dose. So in 2022, the WHO recommended and approved a version of PrEP that can be given as an injection every few months, which will hopefully allow more people to access these medications. And this version isn't quite as effective as a daily oral dose. The injection is about 79% effective at preventing HIV infections versus 99% for the oral version, but still much better than nothing. So we've now reached the end of our lecture. I'll leave you with one final checkpoint to reflect on. What are some misconceptions that people have about HIV/AIDS? And these can be either your own misconceptions or ones that you've heard of that are held by others. Is there anything you learned here in this lecture that challenges those misconceptions?